Welcome back to microbiology. We're going to be talking about metabolism today. I do want to point out that overall, metabolism is going to be all activities inside the cell, including enzymes and what they do, but also, again, the cell respiration, which is something that most people want to focus on, but they really shouldn't, because it's only a portion of what we do in this chapter. So, naturally, with all chapters, we should probably start off by defining what we're actually going to discuss, which is metabolism. Metabolism is basically all chemical reactions that occur within a cell. So whenever I say metabolism, it is literally everything. It is building up of things that we call anabolism or biosynthesis, but it's also breaking things down and releasing energy in things like catabolism. And so to make this simple, we can sum up metabolism in two parts, anabolism and catabolism. Again, with anabolism, we're going to build things up. And to build things, it takes energy, as well as parts. With catabolism, it's the literal opposite. We're going to break things down, which releases energy and parts. So naturally, we have this continual cycle of anabolism and catabolism. They go hand in hand and must happen together. But one thing I want to point out is there cannot be anabolism without preceding catabolism. We can't build anything if we don't have energy or parts. So again, catabolism should always precede anabolism. One thing to point out is ultimately what we'll spend in the form of energy is ATP. And so we like to again convert things like catabolic energy into ATP. But one thing I want to point out is every time you convert the form of energy, you lose some in the form of heat. Okay? And so it's actually a law where, again, every time energy changes form, again, energy is lost as heat. So you can see here the image is illustrating exactly what we just talked about, that, again, catabolism has to precede anabolism, and that over time we can build things up as we break things down. Okay? And so, again, break down resources like sugar to release energy, but also parts that we can use to make other things that we want to later. And to make this simple, really, I'll give you the explanation of what the point of life is. According to biology, life is about reproduction. So everything we're doing in breaking down things for energy and building things up for survival, that all boils down to the simple idea that if I survive, I can reproduce. And in nature, reproduction is king. One of the strategies that developed to continue life was the formation of catalysts. What a catalyst do is speed up rates of reactions, typically by lowering the energy required for the action to occur. And so the idea here is that really these catalysts can be used again and again, decreasing the energy required for a chemical reaction to happen. And as a result, catalysts are incredibly helpful to the reactionary system. And so naturally, catalysts became very dominant. In our cells, and really all cells, we use biologic catalysts that we call enzymes. So enzymes help overcome activation energy. So here's a giant checklist of enzymes characteristics. Again, we'll kind of hit several of these as we go through, but some important ones to point out. Overall, they're made of protein. Again, being protein, they're organic. The goal of a enzyme is to lower the activation energy, thus speeding up the rate of reaction. They are absolutely necessary for life to continue here, and typically do have to physically interact with the things that they're going to break down or build up, but they do not actively become part of the reaction, so they can be recycled or reused again and again. They are affected by environmental features like temperature and pH, so again, they may not function at certain temperatures or pHs. So, how do enzymes work? Well, they actually do work upon a substrate. So a substrate is what the enzyme is going to bind to and literally do something called work. And again, work comes in a variety of forms, but the idea is most enzymes either break things down or build things up. It is important to understand that they physically interact with the substrate, so enzymes physically interact with the substrate, but again, do not become part of the reaction itself in terms of being used. And so the idea is they can be used again and again and again, and that's where we actually gain energy from. The idea is it takes a lot of energy to produce an enzyme, but if an enzyme keeps saving you a little bit of energy every time it's used, as long as it's used again and again, that's potential energy that you save. And so the idea is enzymes make life favorable or more likely to happen. So whenever we talk about the enzyme structure, it's primarily going to be made of a protein molecule. If the enzyme is just a protein, we simply refer to it as a simple enzyme. But many of these are not simple, and in fact are what we call conjugated enzymes or holoenzymes. The idea is we're still going to have a protein portion that we call the apoenzyme, as we can see down here with the purple regions of these enzymes, but we're in addition going to have something like a cofactor or a coenzyme. There are even times where we require both. So the idea here is conjugated enzymes require additional resources to work. So if we provide the resources, the enzymes can do work. But if we don't, the enzymes do not do work. And as a result, we can control enzymatic activity. And the thing to note here is that the cofactors are going to be inorganic and typically going to be metallic. Coenzymes, on the other hand, are typically organic, and again, something we usually can make. So the protein portion of an enzyme should have an active site or catalytic site. This is actually what binds to the substrate. Whenever we bind the substrate to the enzyme, we're now capable of doing work upon it. And so the active site should be designed to fit the substrate at which we're going to bind. This really means that in order for an enzyme to do work, it must physically bind to the exact substrate that it should interact with. There are times where if it's loosely a little off, we'll still typically get work done, but overall, one enzyme is typically designed to do work on one substrate. So what we're really getting at here is this idea that we need a very particular shape of the enzyme, which again falls back to protein folding. There are 4 levels of protein folding: primary, secondary, tertiary, and quaternary. The first level of protein folding is actually going to be the order of the amino acids, as seen here. But the amino acids are going to have hydrogen atoms on them, and thus we're going to start to see interactions creating hydrogen bonds, and that's really the second level. The interaction between the hydrogen bonds create really 2 different types of secondary level folding, which is the alpha helices and the beta pleated sheets, as seen here. But again, typically we need additional features to help further fold, and that's where we get into the tertiary level. But typically, the single polypeptide here is not usually even enough to make a complex enzyme. And so at the quaternary level, we may see additional polypeptides grouped together. So here's an illustration of the enzyme-substrate reactions. In this example, notice our enzyme has a very unique active site that is designed to fit our substrate. And so again, if we don't fit, we don't get work done. So in this example, notice that we bind the substrate and we break it into 2 different parts. And we call that type of reaction a catabolic reaction. So this is a catabolic enzyme. So more on cofactors and coenzymes. We talked a minute ago about this idea that most cofactors are actually metals, things like iron, copper, magnesium, manganese, zinc, cobalt, selenium. There are lots of them, and we usually think of these things as being trace elements. The reason we consider them to be trace elements is these are often going to be recycled, reused, and we don't need a heck of a lot of them because, again, there's not huge amounts of enzymes that require this. And so typically, again, unique enzymes would require unique cofactors that are often metals. Coenzymes, on the other hand, again, being organic, are often going to be produced by the cell and produced at pretty high levels. These things often are going to play a role in the actual process and may even carry things like hydrogens or electrons or carbon dioxides or such. Really, the enzymes in existence fit into 6 different classes or categories: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So let's talk about these 6 categories for a minute. Starting off with the oxidoreductases. This should immediately trigger in your mind oxidation and reduction. If you recall, oxidation and reduction is basically the movement of an electron from one substrate to another, like in one atom to another. And so in this scenario, this must happen together, otherwise the electron can be free, creating a free radical. And in nature, free radicals are quite bad. And so oxidoreductases help facilitate this movement of the electron from one substrate to the other. So naturally, this is a pretty important class of enzymes. Really, dehydrogenases are the same basic concept, because hydrogen is really nothing more than an electron and a proton. You move the electron, potentially the proton often wants to tag along. Transferases are designed to transfer functional groups from one substrate to another. So again, we'll move not core units, but small side functional groups from one substrate to the other. Hydrolases are something we can use to help break bonds by using water. So hydrolases do hydrolysis. Lyases are going to help add or remove groups from double bonded substrates. As you probably remember, double bonds are very strong bonds that require lots of energy to make or break. Isomerases are going to help us create isomers. And honestly, most of you probably don't remember what isomers are, and that's okay. But again, an isomer is going to be something that is essentially going to be the same basic chemical components, but the arrangement is now different. So isomers have the same parts, they're just arranged differently. And so what isomerases do is help rearrange the parts to create a different isomeric form. Ligases help us stick things together. So ligases help make bonds. And if you remember, that is anabolic, and making bonds require energy and parts. So here are some real-world examples. As you can see here, we have some things like penicillinase. Penicillinase is basically going to be a hydrolase that's going to help break down things using water. Specifically, this thing helps break down penicillin. So it breaks down the penicillin, again nullifying its effects. So transfer reactions by enzymes often require the movement of electrons. And so again, that would be accomplished by that group, the oxidoreductases. But we should really again remind you of what oxidation and reduction is. We said it before, but it's important to remember that oxidation is the loss of an electron. Ultimately, that oxidized thing now has a positive charge. But if we move the electron to another substrate, it now gains a negative charge, making that overall charge now reduced or more negative. So again, I don't care how you memorize this, but oxidation and reduction are very important to this class. And so at the end of the day, you should be able to tell me that oxidation is a loss of an electron and reduction is the gain of an electron. We will talk about NAD and FAD in a bit, but really these are going to be coenzymes that help carry, essentially, electrons. Exoenzymes and endoenzymes are pretty straightforward. Exoenzymes are enzymes that are released to the outside of the cell. Endoenzymes, on the other hand, are internally retained and used to do metabolic things inside the cell. Another interesting idea in how we can control how much work an enzyme does is by controlling how many enzymes are there. With constitutive enzymes, you always have the exact same number of enzymes present no matter how much substrate you have. With regulated enzymes, that's the other idea here, that we can increase the amount of enzymes based off of the amount of substrate or decrease the amount of enzymes. Again, more enzymes, more work, fewer enzymes, less work. So let's look at an example of this. Notice with constitutive enzymes, no matter how much substrate is present, very little or a lot, we see the same amount or number of enzymes present. And thus we cannot do any more work than the number of enzymes we have. Over on the regulated side, notice that whenever there are very few substrates, like these yellow popcorn looking things here, we have very few enzymes doing work. But as the concentration of substrate goes up, notice we also increase the concentration of the enzymes, meaning we get to do more work. But what do you think happens if the substrate level goes down? Should we still retain all these different enzymes? Usually not, and as a result, we again see fewer of the enzymes whenever we have fewer substrates. Let's talk for a minute about the role of enzymes produced by bacteria and how they can cause disease for us. Enzymes can actually be secreted out that create situations that cause complications for our immune system or even again our cells. Some good examples include things like, maybe let's start with collagenase. Really, what does collagen do for us? Well, in our cell or in our tissues, collagen is commonly a binding fiber that helps hold things together. But if a bacteria can break down that collagen using collagenase, it can now move from tissue to tissue, and you see how that could be complicated for us. Another example is lipases. Lipases simply break down lipids. But if a bacteria has a lipase, it can start using our lipids as its energy source and increase in number drastically, causing us lots of problems. Enzymes are actually going to be quite sensitive, and they're pretty prone to an action called denaturation. Denaturation is the unfolding or misfolding of proteins whenever they're in situations with high heat, low or high pH, or even certain chemicals. This disruption again changes the shape of the protein, and if you recall, the actual shape of the protein determines what it can do work upon. And so what I'm telling you is denaturation breaks enzymes. And once the enzyme is broken, it can't be used again. And so we have to rebuild them. If we continually break these enzymes, eventually the cell is going to die. That's why you can cook things at lower temperatures that wouldn't straight up kill the cell right away. But if we do enough periodic breakdown of their enzymes, the cell eventually runs out of energy and dies. So that's how you can cook things at low temperature and still kill all the bacteria. So the next thing we should really start getting into is this idea that metabolism is a series of reactions, and these reactions happen typically in multiple steps. And so what we end up with are ultimately these things called pathways or series. And the idea is eventually we'll end up with the product that we want, but we'll go from step to step to step, altering the product from before. And so let's take a look at some examples. So we have three different examples here. The linear pathway, which again would be a line. The cyclic pathway, that again goes in a circle. And then our branch pathways, including divergent and convergent. Divergent means that we separate at one point. Convergent means we come together at a point. So let's go through these. Notice in linear, A gets turned into B, and B gets turned into C, and C gets turned into D. But something to note here is that every single arrow represents an enzyme. There's an enzyme that helped take and convert A into B. But then there's a different enzyme that converts B into C. And then another enzyme that does C into D. So in the cyclic event, notice again as stuff comes in, enzymes take and convert them to new things, and new things, and new things, and new things. But notice at some point, the incoming stuff is combined with the outgoing, and we start the whole process again. The good example of this would be something like the Krebs cycle, which we'll discuss in a bit. In the branch, notice again we typically have that linear that leads to it, but in divergent, notice how N is broken apart into two different products, O and P. But what you should really understand here is this is typically an enzyme that is doing catabolism, right? So the enzyme here is breaking N into O and P. So this is a catabolic example. So what would be going on over here where C and Z get combined to make M? If you answered an anabolic enzyme, you're correct, because we're going to use these two different substrates and combine them to make a single product. So let's talk about competitive inhibition. Competitive inhibition is another way where we see control over whether the enzyme is doing work. With competitive inhibition, really the two words to note here is competition and inhibit. As you're probably pretty aware of what inhibiting means, or to stop something, let's really help define now what is competition. Competition involves multiple parties struggling for the same action or activity, right? And so the idea here with competitive inhibition is you have multiple substrates that want to occupy the same active site. But can we have two different substrates in the same active site at any given time? The answer is typically not. And as a result, that actual enzyme is stuck doing work on just one thing at a time. Non-competitive inhibition, on the other hand, is still inhibiting or stopping the activity of the enzyme, but not through means of competition. What usually happens here is we have a regulator molecule that's going to bind to the enzyme and change its active site. If the active site is not the right shape, will it be able to bind the substrate? No. And as a result, now that enzyme cannot do work. So let's look at examples of both of these. Notice in the competitive inhibition that there are two different substrates that can bind this active site. One of them is the normal substrate that in this situation is broken down into these two products. But notice over here we have a competitor. It has enough of the shape in common with the normal substrate that it can still bind the active site, but the enzyme doesn't know what to do with it. And as a result, the enzyme typically doesn't do anything with it. This blocks then the actual action of the enzyme. Whenever we look at non-competitive inhibition, it's important to note that there are not multiple substrates. Instead, we just have the one substrate, that as long as the active site is the right shape, we can break down into the product. But what's interesting about non-competitive is notice how the product can actually bind to a regulatory site that then changes the active site, creating a situation now where the substrate can no longer bind. And if you recall, if the substrate can't bind, we can't do work upon it. There are additional controls in the enzymatic synthesis line, including repression and induction. Really with repression, we're going to stop something, and induction means turn something on. So let's talk about repression for a minute. Again, if we have more of the enzymes than we really need, it would make a lot of sense to prevent any more enzymes from being produced. With induction, on the other hand, if we clearly see that we don't have enough, it would be understandable to help ramp up or produce more enzymes to get the job done. So you can see the normal pathway in terms of controlling enzymes through repression. So notice here we start off with our DNA that's then transcribed and then translated into protein, and then the protein has to fold to make the enzyme. And notice the enzyme will do its work upon the substrate, producing that product. But a cool thing happens, and that the product can actually feed back and help bind to the DNA, blocking the DNA that is responsible for the production of more enzyme. So we actually stop producing more of the enzyme as we have too much product already. Exergonic and endergonic reactions, again, are trying to illustrate the energy movement. In exergonic, we're going to see a release of energy as the reaction is progressing. The endergonic, on the other hand, is going to require more energy for the process to continue forward. Quite often, again, much like the catabolism and anabolism idea, exergonic and endergonic reactions happen typically together. So let's talk a little bit more about oxidation and reduction. Again, since this is supposed to happen in tandem, we often just refer to this as being a redox reaction. With redox reactions, we see again one substrate is going to donate the electron and the other one will accept the electron. And as we said before, oxidoreductases are enzymes that help facilitate this. Some examples of this would be NAD and FAD that we'll discuss here in just a bit whenever we talk about things like the electron carriers and the respiration pathway. So here's an image illustrating again the redox reaction situation. So as you know here, note here the sodium has one electron in its outermost shell, and it's not real happy about that, so it readily wants to give that up. But whenever you look at the chlorine here, notice it has one too few. So now we have one that's ready to donate the electron and one that readily wants to accept the electron, and that's exactly what happens, resulting in basically ions. Notice again a positively charged ion is a cation, and a negatively charged ion we call an anion. So really the job of NAD is to be a carrier of electrons, and so what we see is the NAD readily wants to accept electrons, but then whenever it does so, it becomes reduced. An important thing to note here is we're really talking about more of movement of hydrogen, and so hydrogen is really nothing more than a proton and an electron, and so if we move the electron, the proton is typically going to want to follow. And so the result is whenever we have reduced NAD, it's NADH with a positive proton as well. So NAD again is very, very important to our process here. It is by far the most common electron carrier, and it does again carry hydrogen and electrons from previous steps to ultimately take them to what we call the electron transport chain. Catabolism is again very important to us. It's our ability to break things down, liberate parts and energy that allows us to later do anabolism. So we'll go through again these steps of catabolism, specifically respiration, in just a minute. Before we do talk about respiration, we should probably talk about what we're trying to make, which is adenosine triphosphate, or ATP. There are really three parts to ATP. The base structure, which is an adenine plus a ribose, which we call an adenosine, and then our three phosphates, phosphate 1, 2, and 3. Naturally, these things don't want to hold on very strongly, these phosphates, and actually hold a lot of potential energy in their bonds. And so by cleaving off one of the phosphates, we can actually release significant amounts of energy. Just as a quick question, what would we end up if we cleaved off one phosphate from ATP? The answer would be ADP, or adenosine diphosphate. Whenever we talk about making ATP, there are multiple different ways we can do it. One of the types we can do is substrate level phosphorylation. That requires really a substrate with a phosphate group, ADP, and an enzyme. And what we'll see is that we can take and help remove this phosphate from the substrate, and then using the enzyme add it to ADP. And the result then will be ATP. Oxidative phosphorylation and photophosphorylation are going to help us produce significant amounts of ATP in a short period of time. Oxidative phosphorylation is a series of redox reactions occurring during the final phase of the respiratory pathway. If you really went into the respiratory pathway at all, it should make sense that the last step is the electron transport chain, where we do significant amounts of phosphorylation and produce lots of ATP. So photophosphorylation is actually going to be the similar idea through which we'll make a lot of ATP, but what's going to drive these reactions is actually sunlight. So this is really the first part of photosynthesis. Whenever we talk about catabolism, there are three major pathways that are catabolic pathways: aerobic respiration, anaerobic respiration, and fermentation. One thing to note here is that glycolysis is going to typically happen in any of these three pathways. So it's the first step and should happen in each of these pathways. So let's show you a comparison now of the three main pathways of catabolism: aerobic respiration, anaerobic respiration, and fermentation. With aerobic respiration and anaerobic respiration, we're going to be doing all the steps, as we can see here. But notice how fermentation is really just glycolysis, and then we have to take the waste products from glycolysis and convert them to other things like alcohols, acids, and gases. So let's talk about differences between aerobic and anaerobic respiration for a minute. Notice where the differences come in here is at the final electron acceptor. Notice down here that the final electron acceptor in aerobic respiration is oxygen from the air. While in anaerobic respiration, notice we need to again have a final electron acceptor like a compound that will readily allow us to take those hydrogen atoms afterwards, like SO4, NO3, and CO3. Aerobic respiration is really a series of reactions that ultimately takes and converts glucose to CO2, and then ultimately allows us to yield lots of ATP by going through the steps. Anaerobic respiration, again, is still going to go through the steps like glycolysis, Krebs, and the electron transport chain, but again, the difference comes as the final electron acceptor. So again, we need an oxidized compound to typically do this. With fermentation, we're going to be typically doing this whenever oxygen is limited or not available. So really this does not require oxygen, but does produce a small amount of ATP by going through glycolysis. So with glycolysis, this is the first step in which we're going to split glucose, but we're going to have a leftover waste product with this in pyruvic acid. And depending on the organism, we'll determine what we do with that pyruvic acid. Some things convert that pyruvic acid into alcohols, some convert it into other forms of acid, or even potentially some waste gases. So we're going to talk about aerobic respiration as being kind of our backbone, because it really is the system that most of us would know and could apply. But we will again branch out and talk about anaerobic respiration in a bit, and then again a little bit on fermentation pathways. So really aerobic respiration is going to be a series of enzyme catalyzed reactions, and ultimately the goal is to take and pull as much energy from glucose as we possibly can. So the way we're going to do this is we're going to go through and we're going to try to track the number of ATPs, we're going to track where the glucose is going, and then we're going to then kind of follow the outputs in terms of what will move on to the next step. So to make this simple, what we need to do is for each of the steps, we need to know what goes in, what comes out, where, and what's happening. So we're going to start this off with glycolysis. And as the name suggests, we're going to take and split glucose. And in doing this, we will again yield a little bit of ATP. Typically, again, 2 ATP is actually gained, and then have the leftover pyruvic acid. So let's go through this process now. Now, if you recall, I told you that for each step you need to know what goes in, what comes out, where, and what's happening. So again, I'll sum up after I go through the process here, those 4 points. But for now, let's kind of go through and show you what's really happening in this process. So notice we start off with a 6-carbon molecule called glucose. That glucose we ultimately want to split. And you've probably heard the saying, it takes money to make money. Well, it takes ATP to make ATP. And so understand that we have to invest energy initially. And so we're going to take and use this ATP and slap a phosphate on the glucose molecule, resulting in glucose 6-phosphate. Once we again have the phosphate on there, notice we can rearrange it to produce fructose 6-phosphate. But this isn't balanced. We again want a balanced molecule to split. So we're going to have to invest more ATP to produce fructose 1,6-diphosphate. Since fructose 1,6-diphosphate is balanced with a phosphate on either end, now we're ready to split it in half, resulting in really 2 parts: DHAP, or dihydroxyacetone phosphate, and then G3P, or glyceraldehyde 3-phosphate. Notice again the reference is, or the difference here is where the phosphate is located. Here on the first carbon with DHAP, here on the third carbon with G3P. So typically, again, we can go either direction, but we'll usually convert it into one or the other. Most of the time we like to convert it into G3P because G3P can be used to make other things later. We call that a precursor molecule because it can be used to make things later. And so let's just follow the path here of G3P, but do understand this happens twice over since we have 2 molecules of it now. So notice G3P, we're going to ultimately add another phosphate to and remove an electron at the same time. NAD is going to be an electron carrier for us. And so notice whenever it comes in, it'll be NAD, and whenever we pick up the electron, it becomes NADH. And so what we have now is diphosphoglyceric acid. And that diphosphoglyceric acid wants to readily give up that phosphate group using an enzyme and substrate level phosphorylation. So we'll take and bring in ADP and add the phosphate to it, making ATP. So let's take and just for a second talk about what has happened here. So initially we've invested 2 ATP, but right here we just produced 2 ATP, right? So right now we are at no gain in terms of ATP. We've met what we invested and nothing more. So really what we need to do through the rest of this is actually take and try to wring out more ATP. And so that's what we do here. Notice the 3-phosphoglyceric acid is going to then be converted to 2-phosphoglyceric acid. We're going to remove water, and we're going to be left with phosphoenol pyruvic acid, or PAP. PEP again readily wants to give up that that phosphate to ADP to make ATP. So now we have a net gain of 2, and we're left with now 2 molecules of pyruvic acid. So whenever we go to summarize glycolysis, we need to talk about what goes in, what comes out, where, and what's happening. So if you remember, to start this all off, we need to have glucose present, as well as 2 ATP that we can invest in helping break down glucose. We also need electron carriers, like NAD, to come in. So what came out? Well, we produced 2 molecules of pyruvic acid. We ended up gaining 2 ATP, and again, 2 NADH ready to carry electrons onto the electron transport chain. Where is this going to happen? Well, in prokaryotes, it's going to be in the cytoplasm. But in eukaryotes, it will also be in the cytoplasm. So what is happening? Well, if we simplify this down, we are splitting the 6-carbon glucose molecule into 2 3-carbon pyruvic acids that will then be carried on so that we can try to gain more electrons and produce more ATP from them. So let's look at what's going to happen with this point right after pyruvic acid. Again, this is kind of an area now where we can branch and do several different things. So pyruvic acid can be continually passed through the rest of the respiratory pathway and again produce ultimately lots of ATP. But if we already have a bunch of ATP, would it make a lot of sense to produce more? And so really this is also an awesome opportunity to start doing anabolic pathways or building things like amino acids, sugars, and fats. But again, if we don't have the final electron acceptor down here in the electron transport chain, then we're going to have to do fermentation, which is again the converting of pyruvic acid into alcohols, acids, and then gases. So the next step is going to be the Krebs cycle. But really we're going to break this portion into 2 parts, the precursor step and then the Krebs cycle proper. And really the goal of the precursor is to prepare pyruvic acid to go in to the Krebs cycle. And so we have to lose a carbon from pyruvic acid and then insert coenzyme A so that we can get into the Krebs cycle. And naturally what we're going to do is again lots of reactions to ultimately take and produce lots of electron carriers like NADH. Again, the remaining carbon from the glucose that we started with will be gotten rid of through this process, and so ultimately we're getting rid of the carbons, we're making a lot of electron carriers, and even in the Krebs cycle a little bit of ATP. So naturally all these reactions are going to happen twice. Since we did have 2 pyruvic acids, again this is going to be done twice over, and so the idea here again, we're going to see it once through, but do recognize it's happening in tandem, and so whenever we total, it's again the twice over because it all came from again 1 molecule of glucose. So we're going to go through both the precursor step, which is this upper portion, and the Krebs cycle all together right here, but again we'll sum up each of these 2 processes in just a minute as separate things. So precursor and then the Krebs cycle. So let's go through the process of chemically what's happening. Again, at the end I want you to know what goes in, what comes out, where, and what's happening, which we'll sum up after this. So let's start off with the precursor. Notice again what comes into the precursor step is going to be a 3-carbon pyruvic acid. But if you notice, what we need to take into the Krebs cycle is just a 2-carbon molecule. So we're going to have to get rid of one of the carbons as CO2. Whenever we do so, we also again get to liberate an electron that ultimately can then be carried later to the electron transport chain. But there's something to draw your attention to with coenzyme A. If you remember, coenzymes are used again to allow enzymes to work. And so in order to even go into the Krebs cycle, we need coenzyme A to be added to that 2-carbon acetyl group, which again creates now acetyl coenzyme A. And with that, now we're ready to go into the Krebs cycle. But do note that this is a cycle that goes round and around, which means the waste product, in this case oxaloacetic acid, will be combined with the incoming acetyl-CoA and then produce our 6-carbon citric acid. So another name for the Krebs cycle is actually the citric acid cycle. And again, it's because we start this process with citric acid. So what we're going to do now is kind of take and ultimately try to ditch the 2 carbons that we brought in so that we're left with this 4-carbon oxaloacetic acid. but in doing so, we're also going to make lots of NADH and FADH2 that will be used later for the electron transport chain. So let's again start off with citric acid. Citric acid is going to be converted to isocitric acid, at which point we're ready to liberate an electron here and liberate CO2, which we can see here. Okay, so the idea is we're going to ditch a carbon, but every time we ditch a carbon, we also make NADH. And what we're left with now is this 5-carbon molecule, alpha-ketogluteric acid. And that alpha-ketogluteric acid, again, we're going to ditch a carbon as CO2, but again, every time we do that, we make NADH. At this point, we're left with succinyl-CoA, but there's still lots of potential energy to be pulled from this. And so we're going to use coenzyme A to allow us to use an enzyme that allows us to make a little bit of ATP. And then what we're left with is this 4-carbon succinic acid. Well, succinic acid we can help liberate electron to allow us to again use later, leaving us with fumaric acid. If we add water to fumaric acid, we have malic acid, and then that malic acid again we can produce NADH from, leaving us with our final product, oxaloacetic acid. But again, since this is a cycle, we combine that oxaloacetic acid with the acetyl-CoA and we produce again citric acid, and we go around again. Do understand again this happens in tandem, so it's not just a one and done thing. We did produce 2 pyruvic acids from glycolysis, so we need to run them both through here to get our maximum potential yield from a molecule of glucose. So let's do the what went in, what came out, where, and what's happening for that precursor. So what came in, well, naturally, since we're doing this twice, we'll do the totals. 2 pyruvic acids will come through here. We also need 2 coenzyme A's and also 2 NAD. What's going to come out? Well, 2 acetyl-coenzyme A's, which go in the Krebs cycle, 2 CO2s, and 2 NADH. Where is this going to happen? Well, in prokaryotes, it's going to happen in the cytoplasm, but in eukaryotes, this is in transition into the mitochondria. And then what is happening? Well, the simplest way to put it is we're taking and removing a carbon from pyruvic acid and adding a coenzyme A to make the acetyl-CoA, which goes into the Krebs cycle. So let's do that summary now for the Krebs cycle. So again, the acetyl-coenzyme A, 2 of them come in. We also need 2 FAD and 6 NAD total. What's going to come out? Well, 4 CO2. We have our 2 FADH2s, 2 ATP, and 6 NADH. So where's this going to happen? Well, it's going to happen in the cytoplasm of prokaryotes, and then in mitochondria of eukaryotes. And lastly, what is happening? Well, really, our job is to get rid of the remaining carbons and glucose as CO2, but at the same time produce as many electron carriers as we can, like NADH2 and FADH, or NADH and FADH2. So the next step would naturally be to do the electron transport chain, or as they're calling it here, the electron transport system. I don't care what you call it. The reality is we're going to pass an electron in a chain to create potential, and that potential will be used ultimately to yield lots and lots of ATP. And so by passing the electron through redox reactions, we're going to be able to push protons across the membrane. And as we get this accumulation of protons, we can ultimately use that to really fuel the production of ATP through an enzyme called ATP synthase. So this is an image that's going to help us understand the electron transport chain. And what's important to understand is what we're bringing in here is NADH and FADH2 that we're again carrying electrons for us. So we're going to actually pass those electrons through these proteins along a chain here. But what's interesting here is every time we pass the electron, we get to move a proton across. And so what we get is this building up of these protons on one side of the membrane versus the other. And again, in nature, that typically isn't okay. And as a result, what we want is for these protons to move back across. And we give them a pathway back across. It's just through an enzyme called ATP synthase. And as those positive charges move back through, it ultimately ends up causing the production of ATP. And so what we end up with down here is a whole lot of hydrogen protons and electrons that can combine together to make hydrogens that then can be used to make things like water. Okay, so an interesting thing to note is we're still going to utilize oxygen to make water, whether it's aerobic like here on the left side or in anaerobic situations here. Okay, so water should always be coming out of this. But again, we get different waste products that can come out of anaerobic respiration, like hydrogen sulfide and things like NO2. So let's summarize the electron transport chain. What went in? 10 NADH and 2 FADH2. And all these were produced before we got to this point. We will again need a source of oxygen so that we can make water. So what comes out? 32 to 34 ATP and water. Again, that range is because of the efficiency. Sometimes we're not as efficient as we could be. Where is this happening? Well, the cell membrane of prokaryotes, because that's the only membrane we have, and clearly we need membrane proteins to pass along. So the cell membrane of prokaryotes. The eukaryotes, it'll be in the mitochondria. Then what is happening? Well, to make it simple, we're going to pass electrons through these electron carriers in the membrane, allowing us to push protons across the membrane. That again creates a potential that's used to then fuel the production of ATP. So here's that illustration of what it looks like in the mitochondria. Again, we still have the proteins used to move the electrons, and again we still get to push the protons to one side of the membrane and again use them to come back through to produce our ATP. So let's talk just for a minute about ATP synthase. It is going to be part of the membrane, and it is an enzyme used to produce ATP. And really what fuels it is that movement of the hydrogen protons through it. And really that whole act of creating potential and then using that potential to make ATP is oxidative phosphorylation, which we again talked about before. So let's talk about chemoosmosis for a minute, because it's really what we're doing by creating that difference in chemicals and really charge here. Concentration gradient actually has a name, and we call that concentration gradient a proton motive force. So the protons motivated or forced the production of ATP through ATP synthase. So the proton motive force, again, is that major disparity between the outside and inside of the membrane. Again, the outside having all the positive charges creates a potential that is used to then fuel the production of ATP. So ATP formation in eukaryotes is going to be the same overall process, but overall efficiency is much lower typically. Many eukaryotes are part of bigger organisms and often don't have to struggle a heck of a lot for food, while every bacteria tends to have to be hyper efficient. So that kind of high-end amount of what we can get out is usually reserved for prokaryotes. Most eukaryotes don't even get close. So here's a nice summary again with the kind of ATP production per step. So again, we produce 2 ATP actually in glycolysis. That is again a gain. We get 2 ATP out of the Krebs cycle, and then we produce the remaining 32 to 34 ATP here, allowing us to get 36 to 38 ATP out. Again, carbon comes in as a 6-carbon molecule, but notice again all the carbon ends up leaving as CO2 by the time we're done with Krebs cycle. And water is, I don't care that much about it. If you really want to care, again, it's really 6 H2Os that would come out as a result of 6 O2s coming in. So we did technically produce 40 ATP, but if you recall, how many did we invest initially? It was 2, right? And so really we only gained 2 ATP from glycolysis. Again, we add additionally the 2 ATP from the Krebs cycle, and then the 32 to 34 ATP from the electron transport chain, and that gets us to our total of our total of 40. But again, since 2 invested, really 38 is the max cap we could have. So again, some more details on the other stuff in respiration. We do again produce 6 CO2 because we had the 6-carbon glucose coming in. We utilize typically 6 O2s and ultimately yield 6 H2Os by the time we're done. But again, the only thing I really want us to keep track of is the fact that all the carbons that came in as glucose left as CO2. So if you didn't pick up how we again got water out, notice the equation down here. We ultimately had half of O2, which is a single oxygen, combined with a hydrogen proton and an electron. So hydrogen proton and electron makes hydrogen. So really 2 hydrogens plus oxygen equals water. So anaerobic respiration, as we stated before, was still going to try to utilize oxygen and still produce water as waste, but we're also going to have additional waste products that come out because we had to have a source of that oxygen. So notice, like in E. coli, they can do anaerobic respiration and take and convert nitrate to nitrite and ultimately water, because again we rob the oxygen from nitrate to use to make water, and we're left with nitrite. And so this is a method, again, still utilizing oxygen as we described before. But something that's important to understand is nitrite is not something that is readily used by other cells. Again, nitrite is not really a usable form of nitrogen, and so we need to reduce it even more down to things like basically nitrous oxide or even nitrogen gas. And so this process is typically done by 2 bacteria, Pseudomonas and Bacillus. And so again, we need them to carry out that recycling of the nitrogen so that we can continue to live. So literally, they're absolutely necessary for us to live. So fermentation is the next thing we're going to kind of summarize here. With fermentation, this is the incomplete breakdown of that glucose. So we still have waste, right? And what did we say the waste of fermentation was? Well, it's really things like alcohols, acids, and gases. So we're going to yield a small amount of ATP, but again, it doesn't require oxygen. So naturally, this is something used in situations where you don't have oxygen. So let's show you a couple pathways. There are tons of these pathways, and they're not really that important for the test, but I do want to kind of point out how we can convert pyruvic acid into things like alcohols and gases and acids. So notice in the middle, we have pyruvic acid. That pyruvic acid can be taken in a couple different directions, and we can take it to the right here and ferment it into lactic acid, but we could also take it to the left here and ultimately take it down to things like alcohols, and sometimes CO2 being produced in that alcohol production. So again, ethyl alcohol has CO2 as being a component associated with that fermentation process as well. So alcoholic fermentation really does occur in yeast and bacteria, but again, drinking alcohol really comes from yeast. Bacteria will produce alcohols that aren't drinking alcohols. Again, never consume anything that is not drinking alcohol, because again, it will hurt you. So I do want to point out really what fermentation is about is trying to free up NAD again, allowing for glycolysis to continue. So the acidic fermentation, and I don't want to go into crazy details, there's lots of variation here, but knowing words like homolactic and heterolactic might be helpful. Again, homolactic acid fermentation produces really just lactic acid, because homo means the same, so we're just going to produce the same product, lactic acid. Heterolactic acid is going to not just produce lactic acid, but also other acids like acetic acid. So again, hetero means different, and so the idea is we're not just producing lactic acid, but also things like acetic acid. So Enterobacteriaceae do have enzymes that can help convert pyruvic acid to several different acids, like acetic, lactic, succinic, formic, and even CO2. And as a result, we typically refer to this as being a mixed acid fermentation. You're probably aware that whenever we don't have sugar, we can basically do catabolism of other things like fats and proteins. So we're going to briefly kind of go through the processes there. Notice lipases are actually going to help break up the fatty acids and the glycerol to allow us to help break down the fatty acids through beta oxidation. So beta oxidation is ultimately going to yield lots of ATP, but there was lots of investment in producing the lipid to begin with. So we do prefer to use glucose if given a chance, but if we have to, we can burn lipids as well. Proteins is a similar idea that usually we don't want to burn proteins. Proteins would be one of the last things we'd want to burn, but we do have mechanisms to burn them. So like proteases help break down the amino acids into their basic components, which then can be plugged in to be used for energy production. Really though, it's important to understand that we really need to produce things as well as break things down, which really leads us to this idea of the word Amphibolism. This idea that at any given point we can either break things down or build things up, and it really is based off the demand of what the cell needs at any given time. And so that understanding that respiration is the typical breakdown pathway, but from there it branches all these other pathways that allow us to break down other things or even build things. So Amphibolism is this idea that we can pass and move throughout the basically backbone, which is respiration, to end up breaking down or building up the things we need is important. And this really illustrates that. Again, the pathway we just went through was taking glucose through glycolysis and the precursor step, Krebs cycle, electron transport chain. But notice we can also break down things like proteins by putting them in as pyruvic acid, or again doing beta oxidation that leads us with acetyl-coenzyme that we can plug in and get energy from. But notice we also have these blue pathways, which are the building up, where we can take like G3P and help make carbohydrates, or even again use pyruvic acid to make amino acids, or acetyl-coenzyme to make fatty acids. So again, we can build or break down based off the need of the cell at any given point. Let's talk about now the precursor molecule idea, like the G3P example we just used. The idea that we can take and at any point kind of pull out and try to do something else with that molecule is that precursor idea. So like G3P being a perfect example. We can continue that breakdown during glycolysis of G3P, but maybe we don't need to ultimately yield more ATP. So let's take the G3P and start building things like carbohydrates instead. And those precursors are what allows us to be able to do that. If we didn't have these branching off points, we couldn't really do anabolism. Pyruvate is another example of a precursor. We can take and do a process called gluconeogenesis. And if you break that word apart, gluco for glucose, neo for new, and genesis is creation. So really what I'm telling you is we showed you this catabolic pathway that went in one direction, but really it can even go in the opposite direction to ultimately allow us to synthesize glucose. And so what we're getting at is this pathway isn't just one direction, that it goes back and forth and branches out in all these different directions. So it's important to understand that really precursors are what this is about, that ATP can be produced or ATP can be utilized to make things that we need. So acetyl-CoA, another example of where we use it to make a lot of amino acids and fatty acids and things like that as well. So DNA and RNA again come from these processes. So really, whenever we sum this up, the backbone of respiration is important, but really everything comes from this process. That is not just one thing. The cell literally lives or dies based off of the choices made through the processes of metabolism. We can naturally make carbohydrates when needed. One thing to always remember with synthesis or biosynthesis or anabolism is that we're going to make things, so we require parts and energy. So it will naturally require some ATP to produce anything, even things like glucose. So proteins, naturally, again, investment of energy, investment of parts, that's going to require resources. But again, those parts are what's going to allow us to make, again, the structures necessary for life. So we are tied to breaking things down and building things up together. And really what we do all this for is ultimately cell division, because as I told you at the beginning, life is really about reproduction. So everything we ultimately do in metabolism is to ultimately yield new cells. And so understand that we break things down for energy and parts, and then we build up things using energy in parts so that we can later divide the cell. So we're going to talk just for a minute about photosynthesis. We're not going to go real far here because, again, photosynthetic organisms don't cause infection, but I do think it's important to at least kind of introduce you to this idea that solar energy could ultimately lead to the production of things like ATP and NADPH, which then can be used later in what we call light-independent reactions to yield things like sugars like glucose. But notice again, plants need solar energy, water, and produce ATP and NADPH that then can later be used to convert CO2 into sugar. So again, plants need sunlight, water, and CO2 and produce waste like oxygen and glucose. But if you recall, what did we use to do aerobic respiration? Well, we had to invest glucose and then ultimately atmospheric oxygen as an electron acceptor. So again, this is a cycle. This is cyclic. Photosynthesizers make the sugars and oxygens that we need, and then we, as again the secondary kind of consumers here, are going to produce waste like water and CO2. So here's that light dependent reaction in a little more specific detail. Don't stress about this. Just know that, again, that first step is about producing ATP and NADPH. And that is then used in the Calvin cycle, which is the light independent reaction, to ultimately produce a whole lot of sugar like glucose. There are both oxygenic and anoxygenic versions of photosynthesis, oxygenic being the one that would produce the oxygen necessary to keep life going, and anoxygenic, again, not producing the oxygen that we would need.